of the Vertebrate Museum at, let's see, it's Poly Humboldt now. Humboldt State University. Just changed its name. So Silvia, this is her first time in the museum, but her lineage goes back deep into the museum's history. She did her she's Brazilian national. She did her undergraduate degree at the Federal University of Espírito Santo in the state just north of Rio de Janeiro in coastal Brazil, where she was mentored by Yuri Leite, who got his degree in the early 2000s, and two other Brazilian students in the museum at that time, Albert Ditchfield and Leonora Costa, were also on the faculty at her undergraduate program. She then went to the Museo Geldi in Belém for a master's degree, and then finally to the American Museum and City College of New York, where she did her PhD. Her major professor then was Rob Voss, very well-known small mammal neotropical systematist. And Rob got his degree at the University of Michigan, where Eileen got her degree, but he was a student of Phil Myers, who got his degree here in the MVZ. Okay, and the last connection with the MVZ that I can mention is that the American Society of Mammalogy set up a fellowship or an award, a monetary award for a Latin American student in the beginning of their career after they finished their PhD. This is the Oliver Pearson Award. It was named after the director of the MVZ, who hired me and retired in the early 2000s and the early 1970s. He taught the first ecology course ever offered in a Latin American university when he was on leave there in 1957 in Buenos Aires, and was later given an honorary degree by the University of Plata in Argentina for that expertise. Anyway, Silvia was I don't know what year it was, but she was the Pearson awardee from the ASM a few years ago, which was a nice chunk of money to help get started. So she's done field work in Amazonia, the Cerrado, and Brazil, and the Atlantic Forest of Brazil, in the wet Andes and eastern Peru. She's now up in Pacific Coast of California, where she arrived in January of 2021. She's just beginning to think about the fauna of the North Coast and of California in general. So hopefully this will be the first of many trips that she'll have down here to the MVZ. So Silvia, we're exceedingly happy to have you here and to listen to talk about her work. Probably mostly, say American, but probably mostly neotropical American.
Yes.
Thank
you, Jim. Thanks, everyone. Got it. So thank you for inviting me, and thank you all for coming. And Jim already introduced me. Let's keep this part. So today I wanted to share a little bit of the research I've been doing, and mostly on neotropical small mammals. Do I have like half an hour? Just let me know. I'm gonna try to be brief because I usually talk too much, but let's see if I can have this delivered in half an hour. Okay, so just to put a context on who I am, what I like, what I do, I kind of consider myself a biodiversity scientist that works with fieldwork exploration and collections-based research for understanding diversity of species, describing species, doing taxonomic work, and also use comparative biology, trying to understand patterns of diversity, including phylogenetic patterns, patterns of morphological diversity, and also patterns of geographic distribution to answer questions in systematics and biogeography and evolution of mammals. And I also work with education and outreach, but today I'm going to focus on research. And I wanted to, the talk is going to be, have two components, main components. I want to share a little bit of my, background and in research interests, and I want to show you one example project on American squirrels and another project which is related to the American squirrels project, but it's not just on squirrels, and it shows a little bit more of a field work, which I bet everybody likes. Me too. So I'm highly interested in understanding which species we have, where they are distributed, their histories, and the processes that might be related to the region and maintenance of this diversity. And we have, an extra diversity, and we still have to learn a lot. Estimates suggest that for each species we know, there are at least 4 species that are still undocumented. And in the tropics, where we have a high diversity of species, this situation is even more urgent. And the neotropical region, which is here, like this, which is here marked in like super high diversity, is one of these leading regions in priority on this rank to be studied, because there is a lot of diversity, but a lot of the diversity is still unknown and not well documented. And the neotropical region contains a great variety of environments and also has complex geological and climatical history, and questions related to species origin and evolution and how that relate to this complex history and environmental complexity are still open. And in my research, I have been exploring diversity and evolution of small mammals and mostly small mammals, and highly focused on neotropical area and more specific in South America. And small mammals, they encompass most mammals. Just rodents are about half of the living mammal species. And mammals, small mammals, they are really challenging because they are still understudied compared to other mammals. And there are like many species, but a lot still to be well documented. So they are a great challenge for mammalogists, just like figuring out how many species, where they are, but they are also a great opportunity for evolutionary biologists to learn about patterns of biodiversity and processes that generate these diversity. So one of the main lines in my research program, I'd say, focus precisely on these most basic fundamental portion of biodiversity science, which is discovering, describing species, and sometimes synonymizing species to like taxonomy. And I consider this important because not knowing the species we have means we cannot have an accurate quantification of biodiversity, which precludes us from any other further questions in ecology, evolution, and conservation. So through the years, I have done a lot of taxonomic and revisionary systematics work. And one important source of new data in biodiversity science and for my research also is fieldwork exploration. And I love doing fieldwork. And that's, just a general thing most people do. And fieldwork data often results in new material that does not apply just to my research, but also compose, natural history collections that will be used broader by the scientific community. And I also base much of my research, obviously, in specimens from museums, which are home for our comparative biology studies. And I collect morphological data. That's one of the main types of data that I use in my research, morphological data, and also genetic data. And more precisely, I use DNA sequences. And for this kind of data, in addition to DNA sequences obtained from preserved tissue samples, which are called modern samples, I also use historic samples, which you might hear ancient DNA too, but which comes from, types of tissues that were not preserved for genetic studies. Skins, museum skins, and crusties adherent to skeletons of specimens. And this has, developed more recently with the advent of new technologies for sequencing, high true output sequencing, and has helped researchers to figure, phylogenetic puzzles and placement of rare and elusive species and so on. And so molecular phylogenies and specific using molecular data, they are really central to my research, to what I do, because phylogenies, or depiction of relationships between organisms, together with, they give me a perspective. I use molecular phylogenies as a background and add morphological data, geographic data, and ecological or behavioral data whenever available to answer questions on systematics, diversification, and trait evolution. And so this is the first example that I wanted to talk about. That's the diversity and evolution of American squirrels, just broadly speaking, but it's more centered on neotropical squirrels. However, these neotropical squirrels, as you're gonna see, they are deeply nested in North American squirrels history. So it's broadly American squirrels. And this project started as my postdoc, 2017. Yes, and at the Smithsonian Center for Conservation Genomics. And I actually started collecting and planning these when I was still doing my PhD. And in 2017, I kick-started the project at the Smithsonian, and I received this other guy, Edson Abreu Jr., which was starting his PhD with the very same title project. And we discovered early on that we were like starting this effort on neotropical squirrels at the very same moment. So he came and he worked with me during my time at the Smithsonian with Jesus Maldonado too there. And we built up this team of North and South American researchers to try to understand a little bit more about South American, Central American neotropical squirrels. So a little background about squirrels. They compose a ecologically important, diverse and globally distributed family of rodents, which has about a little bit more than 300 species and recognized in 5 subfamilies. We have like terrestrial, arboreal forms, we have flying, which are actually gliding squirrels. And among these 5 subfamilies of squirrels, this project frames 2 subfamilies, which is family Sciurini, and specifically the tribe Sciurini within this subfamily, and the subfamily Sciurellini. The first one that I mentioned, Sciurini, includes some of the most common species of tree squirrels that you have around here too. I guess you have like eastern gray squirrel close by. Yes, so these type of squirrels. And the other one is this kind of like a mysterious group of squirrels that includes the single living species illustrated there, Cyrillus pusillus. And apparently this lineage diverged very early in the history of squirrels, but the phylogenetic position of these guys is kind of like controversial. So they are really a little mysterious. And one thing that those 2 groups of squirrels, they share, besides the fact that both inhabit Americas and more specifically South America, is the fact that they have been largely neglected by molecular phylogenetic studies and phylogenetic studies just in general. And for these 3 squirrels, to give an idea, I already mentioned Cyrillini, which the phylogenetic placement of those guys, the pygmy neotropical squirrel, they have been controversial in history, and people really didn't know where they went. And Sciurini, for these guys to have an idea, before the project started, the current project that I'm showing you now started, we had less than 1/3 of the South American squirrels sampled in a phylogeny. So we have very scattered, sample and not a complete, a comprehensive overview of diversity and phylogenetic relationships for these squirrels. And combined to these, comprehensive studies of morphological character variation was also lacking. We had like some point studies like in small groups, but we had no like comprehensive taxonomic reveals, which was really difficult because the diversity is great and we don't have a big like phylogeny to give a perspective and help with that. So there's much disagreement in the literature about how many species or even genera we have, for example, for South American squirrels. So you can see the variation from different taxonomical arrangements in terms of even the number of genera are highly variable. And one thing that these arrangements share is the fact that they were based exclusively in morphological characters, not in molecular data. And as a result of all these, the evolutionary relationships of these squirrels, particularly the neotropical lineages, which were mostly neglected, remain poorly known for a long time, as well as our understanding and consensus on taxonomy, and also understanding on biogeography and evolution. And there is a reason for this special negligence on neotropical tree squirrels mostly, is the fact that different from North America, some North American squirrels, the South American squirrels, they are more elusive and they are mostly trap shy. They don't go on traps, on baited traps, and they are more restricted to preserved environments. And because of that, many species don't have preserved tissue available. So making molecular phylogenies without tissues gets hard, right? But recently, because of the advent of next-gen sequencing and improving technologies, we are able to do that. So for this project, I planned, a long part of the project was on the planning and on the sampling. We did sample very extensively, trying to get historical sample for the squirrels that didn't have preserved tissue to compose a more comprehensive data set that allow us for a more complete phylogeny of the group. And we have used both preserved tissues, our modern samples, historical samples from several, I think 27 different collections from South North America and Europe. And I also did a little bit of field work. We did different field works, and one of these I'm gonna show in the next example project. And after all the sampling and with a relatively complete or as complete as possible taxon sampling, we have used next generation sequencing to target ultra-conserved elements or UCs, which you may know are relatively, they do a good job for both deep nodes because the core of the UCs are highly conserved, and the flanking regions of the UCs, they are more diverse, they have more mutations. So it does a decent job both on deep levels and shallow levels of the phylogeny. And that's why we target UCs for this project. And some highlights is the fact that we were able to sample most of the squirrel species in the tribe Cyrene. And our sample also covers the other subfamily that I mentioned, Cyrulini, which was until then sampled by like 2 only specimens have been ever included in a phylogenetic perspective. So we were able also to include new samples for the rare and elusive neotropical pygmy squirrel. And this was the first result of the sampling effort, which was not EUCs. This was a byproduct. We recovered mitogenomes because our mitochondrial DNA is much more abundant than EUCs. Without targeting them, we were able to retrieve mitochondrial data using bioinformatics. And these mitogenomes data provided, the first result comprehensive phylogeny for these squirrels of the subfamily Sciurini. And this was the first paper from this project. And one conclusion that we reach here with this data set, this mitogenome data set of practical relevance, is the fact that apparently the age of the historical sample or the year where the specimen was collected does not influence the completeness of the mitogenomes recovered. So we did recover complete mitogenomes for specimens that have been collected 100 years ago, as much as we did for specimens collected, in the 70s and 80s, suggesting that you can have great results for mitogenomes, at least, even for old specimens. But there is considerable variation in the success of sequencing, depending on where your sample was stored. And we saw South versus North American collections having a much bigger difference than the age. And samplings coming from South America did a poor job on the results versus North American collection sampling. So we think the quality and the preservation, how can I say, more stable conditions of like humidity and temperature did influence much more the mitogenome recovery than the age of the specimen. And using molecular species delimitation methods together with phylogenetic analysis, we find these 43 operational taxonomic units or autos based on this molecular data for mitogenome data. And we highlight several cases that need taxonomic attention, apparently, based on these molecular data, which I call like red flags on the phylogeny, and includes, possible 6 unnamed species, 3 potential species complex where we probably have more than one species, and some cases of species nested within others that might be synonyms and need to have like a closer taxonomic attention. And we recover several species groups highly supported that we associate to generic names or general names that are available in the literature. And we propose an updated classification at the genus level for these 3 squirrels. And here in this image, each column represents a previous generic arrangement proposed and the names used by previous authors. And then we kind of make use of these other available names for North and South American species. And we restrict Sciurus to Europe and Asia area. Sciurus, before the way it was recognized, as you can see, was not monophyletic at all, as Microsciurus wasn't too. So it's a first step trying to kind of like make a classification team that is monophyletic, and to point particular problems that we have to get new data and more extensive data to try to solve areas of this classification. And particularly for the South American squirrels, although the numbers of genera and species do not differ a lot from the previous taxonomic list or taxonomic arrangement, there are considerable rearrangements and several problems pointed. So there is a lot to be done on taxonomy of neotropical squirrels. And by investigating the evolution of 2 morphological traits that were much used in the taxonomy of South American squirrels, which include the number of the premolars on the upper jaw and the number of pairs of mamma, what we saw is that these characters, they are highly homoplastic. They have evolved several times along the history. So clumping up species because of these in one genus, for example, Microsciurus used to have species commonly small body size with 2 premolars and 3 pairs of mamma. So that was not monophyletic at all. And what we found is, at least in 3 points in the phylogeny, we have Microsciurus interteamed with Sciurus species. So really confusing, but a little bit explain, this non-monophyletic classification, the fact that some of the most used characters are not evolving just once the conditions are homoplastic, right? Which doesn't mean they are not good for taxonomy, but maybe they are not, we can't trust them to put species in the same genus. They might be informative still, and I think they are still informative in a finer level, like having 3 pairs of mama or 4 or 2 can be good for taxonomy, but maybe not for clumping up species in the same genus, because these have evolved homoplastically. And with the phylogenetic perspective, we could also start investigating diversification and biogeography in tree squirrels. And this was the second paper coming out from this project. We have also used the mitogenomic data set, and we did inferred divergence times, estimated ancestral areas, and diversification rates for these 3 squirrels of the tribe Sireni. And we found a higher probability for origin of these 3 squirrels in North America, followed by entrance of the group in Eurasia and Borneo, and then after going to South America. And that has happened possibly during, the Pliocene with the source of South Americans. So notice that invasion from North America to South America and Central America is not one single unidirectional path as previously suggested, but Central America has been invaded multiple times, at least 3 or 4 times along the history, and with source coming from both North and South America. So the history is a little bit more complex than has been, admitted before. And we also report a noded entrance and diversification from North to South America than previously suggested. Previous suggestions had a date of, estimated date of 3 million years for when the squirrels reached South America and diversified. And we found that happened earlier than that, around 6 million years ago, which of course we discussed other potential ways for like how squirrels made from North to South America, even before the Panama Isthmus was completely closed. And our analysis supports some previous suggestions that the South American tree squirrels, they are indeed one of the most rapidly diversifying branches of mammals. And there was a bout of diversification and lineage accumulation after the South American invasion. And it's like shown here, the star, and here, like how we have this like bout of splitting after explosive bout of lineage splitting after the South American entrance. And more recently, last year, we published the results of the larger genomic data set, the UCs, and the ultra-conserved elements that we target. And this data set included, again, most modern and historical samples representing all subfamilies. So with this data set, we were able to answer questions more deep on the phylogeny than with the mitogenome data set, and to try to figure like early splits in the family of the squirrels and where Sayurlini, for example, is. Of practical relevance with this data set, the UCE, is the fact that it provides insights on, again, comparing age of samples. Now, age of the historical samples apparently matters. Different from what we recovered from the mitogenome data set. Earlier, younger samples that were collected not far away ago, they result in more UCs and a higher coverage for each UCs, including the core center UC and also the flanking region, meaning that specimens that were collected a long time ago, they don't perform really well, and they are not, the UCs recovered for those specimens, they are usually just the core center of the UC, and the flanking areas, they don't come together well. So old specimens help with old branches, but not necessarily with newer branches, because the more recent variation is not captured well for these specimens. And if you remember, I said earlier that neotropical pygmy squirrel was kind of like a little mystery, seemed to have diverged really early in the history of squirrels. But now this data set, the UC data set, really recovers a high support for the position, phylogenetic position of these squirrels as sister to the subfamilies Scherini, Calociurini, and Ciurini. And then Hatufini, which is from Asia, is the sister group to our other subfamilies. So we also found deep phylogenetic structure among specimens of the Sayurilini, the living species that I mentioned before, Sayurilus pusillus, in the east and west side of the Amazon, where they are distributed. And just for you to have an idea, the branch lengths subtending these two clades, they are much longer than the branches subtending most congeneric species of the other group, the tree squirrels, suggesting that further taxonomic attention is necessary here. And new dating from these guys suggests that this split was over 5 million years ago. So deep things, like genetic things within the neotropical pygmy squirrel that needs taxonomic attention, likely. And all this information provided by these first phylogenetic studies are expected to have a huge impact on long term on the taxonomy of these squirrels, like by doing a first screen and the red flags on the phylogeny, we can now target like reasonable, treatable problems on the phylogeny, and not just the taxonomy, but we also have shown that these squirrels from the subfamily Sayurini, they experience these explosive bout of lineage diversification after entering South America. Why is that? We don't know. But compared, like this is our data, and previously published data already suggested that South American tree squirrels have rates much higher of diversification than other arboreal squirrels. We don't know why is that. Can we find any correlates for this explosion? That's for next chapters. We need to collect more data for that. So collecting phenotypic data, morphological data, maybe could help with answering this question. And one example branching project of these like first screen that we did for the phylogeny of the tree squirrels is, some of the red flags. Priscilla Seja is starting her dissertation on the Microceurus sabanili flaviventer group, which has apparently at least one unnamed species in the group, and the flaviventer, Microceurus flaviventer species, which is widespread in North South America, seems to be composed by several haplogroups, and also the nuclear DNA also confirms that structure within species. So she's starting to look at the morphology variation and contrasts that with the genetic data that we have to see if we can figure out, is this really a new species? Can phenotypic data support that? And is flaviventer just one thing, or can we have like maybe more than one stuff within flaviventer, because there's great genetic diversity, and there has been also like morphological variation suggested. Up to 8 subspecies have been historically recognized for Microceurus flaviventer. So it's one of the red flags that she's trying to tackle now. And the acquisition of new phenotypic data holds the promise of helping to understand which morphological or ecological features, if any, on these squirrels can be associated with the high rates of diversification in the neotropics. So making the connection, addressing the possible correlates between phylogenetic diversity and phenotypes and ecology, something that would be interesting, not just for these squirrels, but also other squirrels. So we are now starting to work in a collaborative effort to have this phylogenetic screen for the whole tree, the whole squirrels, not just these subfamilies. So in the plans is also like collecting ecological and morphological data to try to answer some of these questions. And last but not, fun, this is the exploration, a project that I wanted to show you, which happened. This project arose in the context of the squirrel project. So when I was starting the squirrel project that I show you, we were missing 3 species that I could not sample. One of these species was this mysterious Peruvian mountain squirrel that was known by one record of like specimens collected 30 years ago on this site in the Peruvian Andes, 1989. And we didn't know much about it, but there was a suggestion from a morphologist, a taxonomist, that the single publication, the single specimen known for this site was a undescribed species of squirrel, and I wanted to sample that. And this place, that's Parque Nacional del Rio Abiseu, and with the study of the potential unnamed squirrel, besides the fact that this place is a World Heritage Site that has really important importance in both a natural and historic perspectives, I wrote a story, a grant, and applied it, and I got fund from National Geographic to go on an exploration to this site. And I gathered a team of people to go targeting the squirrel, of course, but not just the squirrel, because we are going to a very, remote area, difficult to access, expensive to get there. So we decided to do both herbs and mammals in this expedition, target small vertebrates. And this is the team. For mammals with me, it was Pamela Sanchez from Peru and Edson Abreu, which is the other person leading the squirrel project that I mentioned before. And I had one herpetologist, but he went to this expedition as a photographer because we needed images of the squirrel for Nat Geo to, as an argument for the, going on the expedition. And Roberta Murta from Brazil and Renzo Pradel from Peru doing herpetology side of the expedition. I'm going to show you just mammals, okay? But I wanted to share that we didn't go there just for mammals. We were using shared methodologies like pitfall traps that was targeting all those groups. And this happened in late 2018. We went for a total of 22 days in the park, and expedition total time was like 30 days, sampling both small mammals and amphibians and lizards. And we had great success. We collected lots of small mammals that were like, what the heck is that? We didn't know. Yes, a very, very unexplored area. And we visited tropical mountain forest at the northwestern portion of the park, Parque Nacional del Rio Abiseu. And we sampled effectively with our traps open for 15 days. And the range, altitudinal range, was in between 2,600 and 2,800 meters above sea level, which for small mammals is a really big gap of knowledge. We don't have much from like this altitudinal range. So we used, traditional small mammal traps like Sherman traps, Victor traps, and we also used pitfall traps, which was shared between herbs and mammals groups. And we also did active search, okay? And the total number of non-volant small mammals that we registered in the field during our 15 days of sampling was 16 species. And our cubes of like expected diversity suggest that we didn't sample our species. So we expect to have at least 1 or 2 extra species to be collected if we improve sampling time, sampling effort there. And together with previous results from the surveys that happened in the area 30 years ago, the one that collected the squirrel before, that I mentioned. We have plus the 16 species, we have other 7 species registered, with thus sum up 23 species of non-volant small mammals. And on the non-volant small mammals community, a big data set was recently published. They have between 1 and 17 species in over 600 sites searched. And the sites that contain the highest diversity, they have 17 species in 2 sites out of 600 sites investigated. We found 23. This tells us really that we don't know much yet about what we have in the Andes. We have at least 5 species on top of like the most rich sites. So this place is really unique in richness, but it's also a combination of, we don't have a lot of effort, sampling effort in the Andes. We believe truly on that. And to help on identification of species collected, we sequenced DNA and we used barcode site B, cytochrome B. And this was used, to try to access, together with using comparative sequences from GenBank, trying to help IDing the mammals that we have. Some of the mammals we collected, we couldn't even allocate to genus level. We're like, what? That's tentatively Chiptanomis or tentatively Anatomies. And even specialists on the groups sometimes were looking at the pictures of the specimens we brought back, and they were like, I have no idea, this could be that, could be that. So we use DNA because of that. And this is one example species. This is one example species that we collected on the genus Marmosa, Stegomarmosa. There was a single specimen collected of this species, and it's very unique in genetics, according to Site B, and also in morphology when we compare to closely related species. So it seems to be a named species. Another marsupial, which also seems to lack a name. We collected 4 species of marsupials there, and we couldn't find IDs for any. So there are 4 species of SP that we have in our data. The ones that we started to look at, we are from morphological point of view, confirming that they are unique. Some of those are not, are very, very unique morphologically, also, not just genetically. So here's another example, also on genus Marmosa, subgenus Mycorrheus. And the behind the scenes of this photo, which looks, fake, but we spent like 4 hours in a tent with the photographer and 2 people helping. So I should also get the credits for the photo. Yeah, and another example of a rodent now, which is in the genus Dactylomes. And this we were sure about the genus once we collected, but we were like, I don't know the species. The species has 3, the genus has 3 species recognized in the literature. And when we add our sequence, the only specimen collected, and compare that to whatever is available on genome, which is identified as the other 3 species of Dactylomes, we notice that we have a 4th lineage of Dactylomes. So Dactylomes apparently has 4 species. That would be a new species too. And the guy that I mentioned that we didn't know the name, like what genus is that? I have no idea. So that's an ictomine rodent. This is a radiation of rodents that's really special, that's specialized in eating, like they live close to streams and eat aquatic vertebrates. So this we sequenced. We recently had a publication of a big Site B data set for ictomine rodents. And apparently our Chiptanomis, which you call it, like, we're not sure if it's a Chiptanomis, our Chiptanomis is closely related to another weird Chiptanomis that apparently lacks a name too. So we can have here 2 new species that actually also lacks a genus name, because Chiptanomis type is not here, it's here. So the problem is a little bigger than we thought. We didn't know the name of the genus, perhaps because there isn't a name. And the story of this field work is quite exciting because we were collecting all these mammals, and I understand more about morphology of the marsupial. So our new marsupial that we collected, I was really excited. I was like, that doesn't match with anything else. I think it might be new. Very excited. And we were seeing all these other mammals which were not targeted by our field work. You know, the Andean bear, the yellow-tailed woolly monkey, but nothing of the squirrel. We were not seeing the squirrel. And we were like doing active searches, and where's the squirrel? We need the squirrel. Nat Geo wants the squirrel. I needed, you know, to provide a feedback on the squirrel. But, you know, not squirrel, but the bear found our traps, and he was like, "Ow." There was a downside too about having the bear. First day the bear showed up in our camping site. It was the second day that we were camping, and I forgot all the altitude sickness and all the problems that we were having there. It's like, okay, that's worth, we are seeing the Andean bear. And then the Andean bear kept coming back, and then eventually found our traps, and it wasn't that fun. But more of behind the scenes to get these shots, these very shots. That's the photographer. And the bear was really curious. So we were not like going to the bear. The bear was coming, and it was, don't do that, but it was safe. And the thing is we were excited, but we still didn't have the squirrel. So at the very last day of our sampling, when my hopes had vanished already, and we were taking the traps out of our last sampling site, the squirrel made a 10 seconds appearance just to the photographer. That's the only person that saw the squirrel. I didn't. But it was better that the photographer at least could see the squirrel. So this is the only known image of this Peruvian. And Nachi was happy. I mean, the photographer wasn't happy with that. It's my husband, by the way. So he was like, I wasn't prepared to take that picture for like, he was going for a toucanet. So if we have ornithologists here, we saw a very rare toucanet in the area. And then Pedro was going with all his equipment to get a nice photo of the toucanet because the hopes for the squirrel had gone. And then the squirrel showed up. But at least we can tell it's a squirrel, and we were happy. And although we didn't collect samples of the squirrel, we just have the picture. I came back to Peru, Lima, the collections, found the 30 years ago specimens, and collected historical samples. And we were able to place the species in the phylogeny and confirm that is genetically distinct from any other species. So it's also an unnamed species of squirrel. So it was a happy end because at least we had the picture of the squirrel and the story to tell. Taking out the last straps of the last side, the squirrel came. And that's, I promise, my last slide. A little, abstract about our results in this, from this field expedition for small mammals. We have a really high percentage of apparent endemism. All the stars here are the taxa that we identified, the species we identified that are unknown for any other area. So we have 11 out of 16, which is over 70, is like 70%, which I don't believe on that. I mean, I think there is a high endemism in the area, but I think there is a bias. High Andes has not been well sampled yet, and the few sampling that happened didn't necessarily result on sequencing. And this is all based on genetic identification using CITE. So we don't have comparative data from other high Andes areas. And I believe that several of these species will also be distributed in other areas, but we still don't have data from those other areas. So it's showing that's probably, yes, it's a very unique place, high diversity, high endemism, but also pointing that we have big sampling gap, and we actually need more exploration, right, to collect these places, do inventories, and also have data from these places so we can do more decent phylogeographical and zoogeographical evolution investigations about this fauna and yeah, so on. And that's it. So with that, I'd like to thank you. I have a little video here. I hope it plays. Yes. I'd like to thank you for your attention and everybody that helped me, like my collaborators and colleagues and advisors. And thanks for invitation again. And if you have any questions, I will be happy to try to answer. Although I did talk a little too much. It's 1:05. Thank you.